I'm really excited today to welcome my fellow earthworm researcher and friend Victoria Burton to talk about a project that is all about getting communities to look into earthworm communities. So over to you, Victoria, to hear about Earthworm Watch.
Yeah, thanks, Kieran. Yes, yeah, Victoria, and I probably should have done introductory slide to myself. I've got about that. But yes, actually, my day job now is as a researcher at the Natural History Museum in London, which is actually where I did this project as part of my PhD. I should start off by thanking Imperial College and NERC, who were my funders for my PhD, and also the Natural History Museum and Earthwatch Europe and the Earthworm Society of Britain, who were the partners on the project Earthworm Watch. My colleagues there, formerly of Earthwatch, Alan Jones, Anthony Roach, and Jenny Cousins. And at the Natural History Museum, two of my PhD supervisors, Professor Andy Purvis and Dr. Paul Egerton, and also Lucy Robinson, who runs the community science program at the museum. Earthworm Watch, well, firstly, it's a citizen science project, or actually, as we call them now at the museum, community science projects. They're also known as participatory science. So it's actually got very many names, and there's actually literally papers written on the various different names for citizen science, and I'll probably use citizen and community science interchangeably throughout the talk. Lots of kind of discussions on what actually it is. There's not really a clear definition, but it's generally considered to have involved scientific research actually collecting some form of scientific data or other scientific project and engagement with the public, so non-professional scientists, people who are not necessarily trained or paid in science. But it takes many, many different forms. There's ones that are entirely done online. Zooniverse is a good example of a platform like that. So it involves ones where you can just sit at your computer, cataloging, for example, camera trap images, identifying ones like that. iNaturalist is a very well-known one now, which actually kind of bridges both of that, because you could sit online and identify other people's photographs, but you can also go out and take photographs of organisms yourselves and identify them. And all that data out there goes towards scientific research. There's other kind of more well-known ones like the Big Garden Birdwatch, very large mass participation type projects and collecting lots of data, but in a quite structured way, so that they're designed to ask a particular question, such as, what birds are being seen in gardens, how are they populations changing? And similarly, the UK pollinator monitoring scheme is, again, it's been designed to ask a particular question and has quite structured protocols that people go out and record the pollinators in a very protocol way, so they can be compared over time. And similarly, the National Pollinator Monitoring Scheme. So a very, very big variety in terms of the size, the way the structured the protocols, and how simple and how complicated the different ones are, and what sort of people take part in them. Some of them very beginners, just getting to find out about science and how it's done, maybe just want to have fun with their children or grandchildren. Others very kind of experienced people, maybe experts themselves, even though they're not sort of trained in science, what we call sort of amateur naturalists in the past, or expert amateurs. But why did I choose to use citizen science within my PhD? Well, in part it's because as a teenager, I took part in citizen science myself. And during my undergraduate degree, which was at the Open University, they're very one of the earlier universities to use citizen science as a method. They ran the iSpot program, which was another one of the apps and websites that involves taking pictures, put them online and getting people to help you identify them. And they actually had a short module that used iSpot as a tool. So I had to go out and collect observations using it. And so it was quite an inspiration to me as well. But certainly during my experiences as a young person, as a teenager, sometimes I felt I often wasn't perhaps getting the feedback that I really wanted from some of these projects. I'd take part, but then I'd never really hear much about what my results really meant for science, what kind of the answers were, you know. And that was always a little bit disappointing for me as someone who was very into science. And part of me thought, maybe I could do better. But also there was a very good practical reason. My wider PhD was looking at the effect of land use on soil biodiversity, and several of my chapters was collating existing projects that had looked at that topic. So I'd go out, I'd find papers I published on that, collate the data together. But comparing those that had looked at above ground biodiversity and soil biodiversity in different land uses, it was always fewer that looked at soil biodiversity. It is sadly less well studied than above ground, but particularly in the urban areas, it was much less studied. So I had just over 1,000 sites that had studied above ground biodiversity in urban land uses. This is across the world, but I'd only managed to find 359 which had looked at soil biodiversity. And this was kind of an issue for me in my analysis, because I wanted to compare above and below ground biodiversity to create my models of how they're affecting and the comparisons between them. And also, although it's a relatively small amount of the UK, in this example, so 8% of the land use is urban, quite a large number of people, I think it's around 80% live in urban areas. So it's really important that we have healthy soils, and soil biodiversity is a major part of what keeps the soils healthy. Recycling the nutrients, important in drainage, for example. But of course, with citizen science, it relies on people collecting the data. So you automatically get a bias from areas where there's more people, such as urban areas. So I thought citizen science would be a really nice complementary data set to those ones that I've been collecting. So hopefully win-win. And of course, it's a really nice way of involving the public in soil science and soil biodiversity. So this area that perhaps is overlooked compared to a lot of above ground biodiversity. So soil animals, as I said, maybe perhaps underlooked, and one example of this as a comparison is the ladybird survey of the UK. So ladybirds, they're above ground, they're quite brightly coloured, although not all of them. So there's 46 species of ladybirds in the UK, but only around 26 of them are kind of the ones you might actually associate as being ladybirds, are sort of red with or other bright colours with the dark spots. And the ladybird survey kind of started around 2005 in response to the invasive harlequin ladybird. And this shows the records collected from there to 2022. So this is from the NBN Atlas. As you can see, there's over 123,000 records collected, and a pretty good spread across the UK. There's gaps in the less well populated areas. But if you compare with the not dissimilar number of species of earthworms, 31 species in the UK, and started not a little bit later. I mean, I've put in brackets here that the actual National Earthworm Recording Scheme started 2014, but the actual Earthworm Society started 2009, so a little bit later than the ladybird survey. Much fewer records and much sparser, perhaps even more strikingly, much fewer number across the UK. And we know, they're just as common, but they're a lot harder to find. You've got to dig them up, and they're maybe not as obvious, not as sort of brightly coloured, maybe not as charismatic. Well, yeah, I think they are. Yeah, so yeah, it's quite striking the difference between those two different groups, even though they've got a similar kind of number of species. But despite that, there have been earthworm citizen science projects around the world. So this is a little review that myself and colleague Erin Cameron did for a really lovely paper that we wrote for Frontiers for Young Minds. So it was part of a collection of papers we did, colleagues and I. So Frontiers for Young Minds is a journal that's actually aimed at children and young people. And they're actually papers are submitted by scientists and reviewed by young people. So the young people and the mentor actually read through the paper and give the scientists comments to make sure they're accessible and understandable young people. And it was one of the most fun things I'd done, getting comments back from young people. And so this paper was sort of an introduction to what citizen science is and some of the earthworm projects out there. So as part of that, we went through and looked at some of the earthworm projects, past and present. I think the oldest one we could find, if you discount the work that Darwin did, which could arguably be citizen science. He did get a lot of his friends to send him earthworms, study earthworms. Was a project a while back called Earthworms Down Under in Australia. But there's been quite a number of other ones as well, notably in the United States and Canada, which are mostly actually to do with earthworms as an invasive species, because a lot of them there are introduced from Europe and they're not native there and actually have a negative effect on the forests there. But there's also been quite a few in Europe. So that's quite a nice little look there. We're trying to finding out some of the ones there. And they've been aimed at various people, a few at farmers, and quite a lot at school students. And probably most significant one in terms of inspiring and influencing the development of Earthworm Watch is the Opal soil and earthworm survey, which my supervisor Paul Eggleton was also involved in, and Lucy Robinson as well. So it was a very large program run by Imperial College, and there was actually a number of different surveys run as part of Opal. And the soil and earthworm survey was aiming to look at soil health, I think particularly looking at again urban areas and pollution as well. So it ran for quite a long time, started in 2009 and finally wound up in 2020. So during that time, it had over 5,500 responses. And it actually aimed to try and identify the earthworms to species using a flowchart guide, and also collected up soil properties as well. So looking at the moisture and the soil type and whether there was any presence of sort of foreign things in the soil, such as bricks, because there was that element of soil health there as well, and had a good spread of data collection over that time because of the long time it was up to. This is up to 2015. There was also some quality control projects around it as well to check, how well often young people were identifying it. There was a big aim in, because it was funded by a lottery grant, there was a lot of emphasis on getting disadvantaged communities involved in it. And unfortunately, they found, although identifying to sort of the major groups of earthworms was fairly successful, so 88% were actually identified correctly when they took that sample of ones that identified and then had them identified by an expert. Only 60% of the actual species were identified, and that varied from species to species. So some were okay, but others actually, none of them were identified correctly. So knowing this from the previous work, we thought, there's no point trying to do that again. We're trying to get within our project identifying to species, because we know it doesn't work very well. No point redoing that. But 88% for ecotypes, that's reasonably okay within the kind of acceptable error range. So for Earthworm Watch, we decided to use that same approach, separating the earthworms into 3 kind of main ecotypes that you find within the soil. So in case you don't already know, if you've been to some of Kieran's talks he's hosted before, you might already know. So we called them for the project surface feeding. So this is what scientifically is the epigeic species. They tend to be fairly small and dark red coloured, living within the surface of the soil, feeding on leaf litter, and don't make permanent burrows. And then the soil feeding ones we called the endogeics, which live in the kind of top 20 centimetres or so of the soil, making horizontal burrows and feeding on the soil itself and sort of digesting out any organic particles in there. And then finally, what we termed the deep living earthworms. So these are the Nesix ones, can get very large. Said over 15 centimeters was the cutoff we used in our guide. Can make very, very deep burrows, and often vertical burrows, which they live in permanently, popping their heads up at night to collect leaf matter, which they drag in and that they feed in. Primarily feed on leaf litter. They do eat some soil as well. So that was our main decision using the ecotypes. We had to decide kind of what was our main aims. So obviously they were aligned with my overall thesis of looking at how soil biodiversity is affected by land use. But the main aims here were to kind of map the overall earthworm abundance and the kind of ecotype diversities, and find out which urban habitats kind of earthworms prefer. So the focus here was on urban, and find out sort of within that the earthworm's response to environmental factors and how earthworms are related to soil carbon. So Earthwatch Institute had particular interest in soil carbon. It's Alan Jones's particular research interest. So we wanted to measure as a proxy the soil carbon and then look at the earthworm numbers of that. And of course, importantly as well, engage the public in soil ecology. So important difference with the Earthworm Watch survey is how we designed it. So of course, already mentioned one of the problems with the previous citizen science projects was identification, but there are others as well. People obviously vary in how well they do the protocol, depending on their own ability or just with how seriously they're taking it. But there's also because it's going to vary by when they do the survey or where they are in the country. There's lots and lots of different variations. So approach we used was to use a paired sites to try and filter out some of those problems. So we asked people to survey 2 different habitats, for example, as in this picture, a flower bed or lawn, but they could also do a woodland or a vegetable patch. We gave them a list of options, and or they could do a different management type as well. So if they had an area that they fertilized or didn't fertilize, they could compare that too. And then particular statistical technique I was going to use called mixed effect models actually helped kind of take out some of that variation and actually try to statistically fix some of those problems with the citizen science data. Not all of them, but hopefully try and allow for some of those problems. So we actually used a standard earthworm sampling method, very familiar to anyone who's done earthworm sampling. Dig out a set size hole, look through the soil, pick out the worms into a little tub, and then make up a solution of mustard powder, hot mustard, and just like Coleman's mustard powder, into water and pour it into the hole. So it's just a light irritant, tickles them, brings up any of these deep living earthworms, which can live much deeper in the soil. And hopefully if they're there, they come up for counting. Doesn't always work, of course. Sometimes they have exit burrows and go out, but we did do some checking and found if you don't use the mustard, you do underestimate some of these big anisic earthworms. So counting identification, we provided a chart to help the participants separate out these different ecotopes, these really, really big anisic worms, the surface feeding epigeics, and the paler entoGeic soil feeders there, based on the colour and size. Not using a key, just using a guide here and a ruler. So asking if they're more than 15 centimetres long, reddish brown in colour, or less than 15 centimetres long, reddish brown in colour, or if they're pale and pink. There will be some overlap between this, especially with the juvenile deep living earthworms. But actually there's some evidence that the deep living earthworms act more like epigeics where they're juvenile anyway. Oh yes, we also asked them to separate them into adults or immatures using the presence of the saddle as well. And we also asked them to look at the different soil properties as well, because obviously we want to look at the environmental differences. Used a very simple soil texture flow diagram to separate them into sandy, loam and clay soils, and also the soil colour chart to separate them into different hues and shades, and this was the proxy for soil carbon. So the darker the soil, roughly, the more carbon there's in it. So it's the soil texture. It's quite fun to do, roll it into different shapes. And soil moisture we also just did on a very simple, if you can squeeze it, and if there's water runs out, it's wet. If it holds together, it's moist, and if it doesn't, it's dry. We also provided a little sachet of vinegar to pour on. Again, it's very coarse. If it fizzes, you've got calcium carbonate in it, which we know that earthworms tend to be more numerous in very chalky soils. Here's our colour chart. And we also asked for how much plants were covering the square before they dug it out, whether it was completely covered, partly covered, or no plants at all. So fairly simple environmental properties. When we piloted, we did try some more detailed ones, actually giving out test kits to check kind of more detailed ones, but the feedback that it took too long, it was too fiddly. So we had to dial it right back. And that's one of the compromises you often have to do with science in general, but particularly with citizen science. If you make it so complicated that no one wants to do it, then yeah, you're not going to get your data you want. So we went live in April 2016. It's an exciting day after a lot of work went into it. This was the pack that we sent out. So participants registered on the website and they could either be sent the pack out. So it came with their little packets of mustard and vinegar and their booklet and their chart. Or if they had their own mustard and vinegar, they could actually just get on and do it themselves. They could download these. And we had lots of events as well to try and get the word out and engage people. This is one we had at the London Wetland Centre. It was really, really fun. We had lovely weather, and Earthwatch and the museum staff got there. Also, I got to see a really nice worm that lives in wet ground and I hadn't seen before, so that was exciting. We did some earthworm charming just about to see the grids in the background. It was good fun. And another one, I went to the Bristol Festival in Nature, which was a bit more static. There, talking to people about worms, had some the different ecotypes, and it got people to try and identify them there, and a little fun competition for people. And some of these were sponsored by the British Ecological Society as well. I applied for some funds to go to them, so it was really nice. And unusually, I think for a PhD project, it meant I got to do lots of really nice, fun, different things. So I got to do some talks at the museum in their Attenborough studio, write articles for the museum's magazine, do some little articles for different newspapers. I had a blog on the Guardian's gardening blog, which was great. Little interview for local television. And I got to go on Gardener's Quest in time on BBC Radio 4, which was always on my bucket list to go on BBC Radio 4. So that was really, really great. As lots of really good fun things, which is really lovely to get out there more, not just sit at my desk crunching and reading and writing. So, and also really, really exciting that the British Ecological Society awarded me a public engagement award for the project, along with a couple of other colleagues. Yes, I got to go to award ceremony and go up on stage, and yeah, and got a dinner and wine. So that was lovely, and yeah, really, really exciting. So who actually took part? So we ran between April 2016 to August 2018. Decided to kind of wrap it up after that time. We didn't have the kind of funds or capacity to keep going, and sometimes, you know, it's good to kind of call it an A and wrap up and give out your results and get written up. So we had 1,678 register, and of those 214 completed at least one survey, and actually seems really small amount, but it's actually comparable to this type of citizen science survey. Most people do not do the survey, and it's actually very similar to the Opal surveys and to others that have recorded them. A lot of people don't actually know how many don't take part. Because we actually ask people to sign up, we have that information, which is quite unusual. We had estimated over 7,000 actually engaged in these events, and these are some of the lovely tweets that I received, people taking part, which was always a joy to see. So I haven't got round to finishing my paper on the participants and who took part, but hopefully I'll get it finished really soon. One of the most interesting things I found about the registrations that we had much more women registering than men, which is more, most of the work you find out there says that citizen science is dominated by men, but there's also some others that say it's not. So it seems a bit variable, but that was quite interesting finding for me. And you can see it's a fairly big age range, but we do have a peak around here. But this is those that registered, not who took part, because we weren't able to record that. And we also asked why they wanted to take part, which has been an interesting data set that I've been analysing, but haven't finished writing up yet. And I've been categorising it into different, what they call motivations. And people have categorised kind of these main reasons for why people take part in citizen science. There's enhancement, so it's like they want to kind of improve themselves. So this is an example here. They said they wanted to feel useful. There's career ones, so if they want to kind of improve their career prospects, sort of understanding, they want to learn more about earthworms or soil or science. Sort of social ones, how they just sort of want to get out and do something fun with their children or grandchildren, friends, and values. So ones that are worried about nature, want to make a difference. And so I went through all the reasons that people are given and classified them, because some fell into more than one of these categories, and plotted them out. And they also fit into kind of extrinsic ones, so ones that are kind of more about others, or others intrinsic ones, so more about their kind of values. And so those actually the top ones were sort of understanding, wanting to learn more about either earthworms or soil or science, and others, and the second top one was values, so they wanted to do something for nature. And these actually consistently, generally, when people have looked at motivations, tend to come out with the citizen science ones, and sort of much less on things like social and enhancement, and career is very small indeed. So this is actually for the museum is quite interesting, because we haven't always looked at this sort of information. So I need to get a move on and try and finish writing it up and communicating it out, and sort of think about, you know, who we're trying to reach in these projects, and how we actually tailor our messaging. But I have managed to publish all the results on the soil and earthworms, and there's some links here. So firstly, the actual paper. So that was published last year in the European Journal of Soil Biology. It was really important to me that that was open access, because of course it was collected by the public, and it was important to me that everyone should be able to access it. So that is open access. But of course, it's quite an academic read, and not for everyone. So the museum also, alongside that, has an article on the results, which can be accessed there as well. So there's kind of the results highlights and a news article as well. And also the actual data set as well is also publicly available. It's Creative Commons license as well. So if anyone wants to do any further work on it or use it in any way, it's available there as well, if anyone wants to access it. I think that's important, because it was collected by public and it should be available for their use. So the results, so we had 652 complete surveys in the end, just over 5,170 earthworms counted in total. Reasonable spread across the country, the usual areas, thin, the usual southeast bias there. It's not terribly surprising, given it was museum and Oxford were the major partners. So the average number of earthworms per pit was 8, and that varied between 0. So actually 11% of the soil pits didn't have any earthworms in at all, and a whopping 102 in one of them. And so that actually worked out to be about 200 per square metre. So in terms of my modelling, the way I put it in is kind of the main parameters. So you start off by popping in everything, and then running it and gradually dropping out the terms which are not significant, until you're left which was known as the best model. So I popped in the land use, soil texture, moisture, fertilizer. So this is the fizz test, whether it's got calcium carbonate, the darkness, and the plant cover. So, and these are the terms which I put in to kind of account for those participant effects. So these are what we call the random terms. So this is what the study is, and this is the pit ID here. And I've also put in these other what we call smooth terms. So these are where the location of the survey was, and when it was in relation to the start of the term survey. So this just sort of cancels out any differences in the time and the location of the survey. So it just kind of accounts for some of the variability according to that. So we're just kind of trying to focus on what we're interested in. So the land use and the soil properties. So once I've done the modelling, we've accounted for these effects of the study and the pits. We found location dropped out, it didn't matter. Time was a significant. That's all right. That's helping to filter that out. So we're left with the bits that we're really interested in is come out as significant is soil moisture and fertilizer. But first, let's look at how the abundance was across the whole of the UK. This is the average per soil pit. There is a bit of a caveat here because you can't really put it across all the other habitats. This really only applies to urban habitats. And you can see this, it seems to be slightly more earthworms in this sort of part of the UK, possibly kind of more earthworm friendly soils there. But again, it's only really applicable to urban habitats. So I think it's a bit limited what we can really say. Habitat actually wasn't as important as the soil properties. So it's only the P equals 0.015 means it's only so it is arbitrary. But in science, we usually say if it's more than 0.05, we say it's statistically significant. So this is actually really borderline. But one really interesting thing about this, and you can see, well, the earthworms didn't really have much difference. So this is the number of earthworms per pit, and these are the error bars. And you can see the error bars are much the same. They don't really seem to mind what habitat it is, apart from flower beds. These are much, much lower number of earthworms per soil pit than all of these habitats. And this was really interesting because another citizen science survey by Martay and Pierce Higgins of the British Trust for Ornithology, which is looking particularly at earthworms in school sites, also found particularly low numbers in flower beds, which is like, oh, that's really curious that they found a similar result. And perhaps it's because flower beds are really often dug up, and we know that earthworms don't like that much as soil disturbance, but that was just very curious that they found that similar result. So moisture, this is highly significant. It's actually, really, really significant. And it's not too surprising that we found the most numbers of earthworms in these moist soils. Not keen on dry soils, not keen on very wet soils, like the nice moist middle soils. And similarly with the soil texture, highly significant result, particularly in the sandy soils, much lower numbers of earthworms, significantly lower numbers of earthworms. They really didn't like these sandy soils. And something a little bit more interesting was the fertilizer result. After our first kind of year, we were finding a lot more earthworms in the vegetable beds. We thought, is this a true thing, or is it because they're more fertilized? So we added another question about if they fertilized and what type of fertilizer it was. Inorganic, whether they don't have fertilizer at all, organic fertilizers. And interestingly, we found there's more earthworms, significantly more, the star is, earthworms in those that are fertilized with organic fertilizers, but not when they fertilize with inorganic fertilizers. So ones that had used things like chicken manures or farmyard manures, compost, things like that, but not when they had used inorganic pellets or sprays and things like that. So what about the ecotypes? So for this model, I used split it out into the different numbers of ecotypes, use the same different parameters, but with an additional one, which is called an interactive effect between the ecotypes and the land use, because I wanted to know if the ecotypes were responding differently to different land uses. And then I use these same terms to just kind of control for those location, time, and the studies again. So this time, only the fizz tests, the calcium carbonate dropped out. So a lot more was in the model, a lot more complicated model. So the main takeaway with it was that the interaction between the ecotype and the habitat was highly significant. So these different ecotypes were having different responses to the land use. So they were not all responding the same way. And kind of the main takeaways from those were these endogen, the soil ones, which were in 71% of the soil pits. We saw more of them in these lawns, grassland habitats, more in the vegetable beds, and actually fewer in the woodland habitats. And the epigeics, on the other hand, we saw 62% of them in the pits. They have more of them in the hedges and bushes habitats. They were at the highest numbers in there. And the anisics, they're only in 33% of the pits. That's quite typical. They tend to be the lowest numbers of the earthworm groups. But they were highest in the lawns, the meadows habitat, and also, again, in the vegetable beds. And one of the final questions that we want to answer was whether actually this paired approach makes a difference. That was quite a good thing. Quite, it was quite a novel thing. We wanted to know whether it makes a difference. Actually, that proved to be quite a difficult thing to find out. It turns out that the method I was using, no one had done the mathematics to find, to work out how to extract and calculate properly the difference that makes. And because I'm not a statistician, I don't have the kind of ability to do it myself. So I had to use a different method, which wasn't quite doing what I wanted, but it had to be good enough. So this is only an estimate, but roughly the paired method accounted for about 30% of the variation for the total earthworm model, and about 60% of the variation from the ecotype model. So it certainly seems like doing this paired approach does remove a fair amount of that variation between the from the models. So it certainly seems that it's worthwhile. It's one of the nice things about making my data available. Perhaps someone who is more statistically minded might be able to do some other analysis on it to actually kind of perhaps to do some other analysis. What do they call it? I can't think of the word right off my head, but to simulation, that's it. To actually find out more about how much of a difference it actually makes. So what does that actually mean in terms of earthworms and managing our urban areas? Well, hopefully it provides some clues on how to manage our spaces better from earthworms. Certainly, it's certainly more evidence that using organic compost, such as manure and homemade compost, are better than inorganic ones. And that, again, that sort of another project showing that earthworms are not as numerous in flower beds, perhaps is evidence that disturbing the soil regularly is not good for them, and sort of no dig methods, mulching and such, is probably preferred. Perhaps having permanent planting instead of flower beds that are reused every year would be better for urban maintenance, perhaps. And leave the leaves. So the elisic and the epigeic ones, they feed on the leaves, and we found more epigeics in these hedges and bushes, which obviously producing leaves, and they provide food, but they also provide habitat for those earthworms. And of course, have a variety of garden habitats. We're finding different ecotypes in different habitats. So if you can provide more habitats, you can provide for those ecotypes, and hopefully also that if that's a proxy for the different species, you're also providing for different species. So yeah, I think I'll close it there and head over to questions.